realize it was the first webinar of the series, so it puts a lot of pressure on me, Meng Meng. If I do a terrible job, no one will come back for the rest of them. So, but I want to thank you. As Meng Meng said, I'm down here in College Station, actually about a 10-minute walk from where Meng Meng is in the horticultural building. So I'm in the entomology building, and I really appreciate the opportunity to give this webinar. So insect vector diseases have received a lot of attention recently. Obviously, we've heard of things like West Nile and citrus greening and Zika. So some of the, I think, very interesting diseases are in plants. And so today I'll talk a little bit about thrips vector diseases. I'll focus mainly on the group called the Tospoviruses. This is probably the main group that are vectored by thrips. If you're not too familiar with thrips, I'll talk a little bit about them as well. One thing to note is that there's actually quite a few different Tospoviruses, but the one you've probably all heard of is the tomato spotted wilt virus. That was actually the first one that was described, and for a long time was thought to be the only one in that family. And these viruses are an interesting family in the sense that most of the viruses in the family are actually animal viruses, infect animals. So this is interesting that this particular group has found a way to infect plants. So as you would expect, tomato spotted wilt virus is mainly a big issue in tomatoes, but also impacts a lot of ornamental plants. So I'll talk a little bit about some of the ornamental plants that are impacted by not just TSWV, but the other Tospoviruses as well. So in the case of ornamental plants, it's really mainly a production issue. These viruses do occur in the environment, but really it's when you have production houses, propagation, nursery production with a lot of plants that you tend to get issues. And one of the, you got a little animation there, one of the things that to be aware of is that this group of viruses are potentially on the rise for reasons I'll talk about, and yet still a lot of growers are not necessarily aware of them. So I'm hopeful that this podcast, I'm hopeful that this webinar will help highlight some of the issues and will be of some help. Okay, so here you have a very clear symptom of one of the Tospoviruses. This is the impatient necrotic spot virus, or INSV, which is probably the second most common Tospovirus that we have in North America. And this is on Gloxinia, and this is a very susceptible host. So these viruses really impact a lot of bedding plants, a lot of crops produced for hanging baskets, and so forth. So here we can see Gloxinia is very susceptible. When plants get infected when they're young, one of the symptoms you'll see is stunting, and this is obviously a very clear symptom here that you can see. Okay, so obviously before the virus can infect the plant, it has to be spread by the vector. So I'll just talk a little bit about the vector and some of the symptoms to look for. So this is probably the most common vector that for Tospoviruses that we have. It's not the only one. I'll talk about some of the other ones. Many of you are probably familiar with this. This is a global pest that's traded and shipped internationally in plant materials. It's a very small insect. It's about 2 millimeters long, or a 20th of an inch. Thrips generally, certainly most of the thrips that spread these viruses are pale brown, actually yellow to dark brown, but mostly pale brown. They have these fringed wings that you can see in the picture here, which is where they get their name from. They're very slender, they're very cryptic. They have the ability to reproduce quite rapidly and develop quite large numbers. And so they can be hard to spot, but, and this is where it gets depressed. Oh, no, the next slide is the depressing slide. Okay. So in addition to vectoring the viruses, these thrips also cause damage through just direct feeding damage. They have what we call a piercing and rasping feeding behavior, similar but not the same as aphids. They actually destroy the cells as part of their feeding. They don't tap into the phloem, but they do cause this kind of speckling and spotting damage, which looks a little bit like spider mite damage superficially. Many of these thrips that are virus vectors are also flower feeders. So you can often get symptoms on flowers. And these would be fresh symptoms here. You can see this kind of pale speckling. Now this will bronze up over time. So if you're looking at older damage, it will often be darker. And also if the damage occurs when the plant's still young, you also tend to get a lot of deformation as well. So these are the kind of symptoms to look for. Okay, yeah, this is the depressing slide. So one of the issues with thrips is they're really very hard group to tell apart. And given that different species can actually vector different viruses, sometimes certainly from a biosecurity perspective, we want to know what species we have, certainly when it comes to detecting shipments from internationally. Oftentimes the only way you can get a positive identification is through slide mounts. You can see here, these are 3 other common vectors of Tospoviruses in the United States. Tobacco thrips on the right, thrips tabasi, which is onion thrips, and the one in the middle, which is another related to the western flower thrips. This is actually a common blossom thrips. This is quite common on tomatoes. It's common in Florida. I'm not sure how common it is here, but they're really hard to tell apart. So oftentimes this just doesn't happen. You know, we don't always necessarily know which species we're dealing with unless this diagnosis is made. Okay, an easy way to sample for thrips is just simply to use some kind of beading tray or use some kind of white paper or an artist palette can be used, and you can simply tap the flowers or the growing portions of the plant sharply, and the thrips will often be dislodged. And you can see here some thrips on the paper there. So you're really looking for small, pale brown, slender insects about 1/20 of an inch long. The larvae look a little bit different. I'll show you what the larvae look like now, but probably any grower is familiar with what thrips look like and probably just doesn't want to see them. Okay, so I do have a short video here which I will play just so you can see the difference between the adult and the larval stages. So I don't know whether you can see my marker, but here you have an adult on the upper left portion. You can see that kind of dark line down its back is its wing line where the wings are folded down its back. And there's another adult running up and down. And you have a larval stage in the middle. The larval stages are pale yellow. They don't have the wings. And anytime you're sampling and you have larvae, that's indication that you have a growing population, that they're starting to reproduce and that the population is already growing, because it takes about a week after infestation when you'll start seeing the larvae. Okay? Okay, I just want to talk a little bit about the disease cycle for Tospoviruses. I think it's important to understand where they come from and how they start an infection. This can really help with the management. So now these viruses are not persistent in plants in the sense they can't be transmitted through seed. So anytime you have a disease incident, it must have come in on a live insect. And so in the case of thrips, what commonly happens is that in the winter or during the non-crop cycle, these thrips occur in the environment. They often feed on weeds, which can serve as hosts for the virus. There's many different weeds, but many weeds in the nightshade family are commonly hosts, but really very many. It's only the first stage of the thrips, it's only the larvae that can actually acquire the virus. But typically it's the adult which transmits the virus because that's the stage that's mobile and can fly and infest a crop. So typically in the spring is when thrips become active and start reproducing, and you get large dispersal. And so that's an important time to look out for them. And you can see here how they're moving. In this case, this is a tomato crop, but it could equally be a nursery. And once the viroid thrips are in a crop, they can actually reproduce potentially, and their progeny can also become infected in the crop and spread the disease. That's called a secondary or secondary cycling of the virus. Typically what research shows is that in most cases, most of the issues is caused by the primary spread, which is the thrips that are actually coming in from the environment and spreading the disease. Oh yeah, I just put this in because I knew I would forget. So there's some additional routes of infection as well. It's not just immigrating insects. You can also have other additional routes can be, thrips can also be brought in on infested plant material. You can also have propagation of plants. If you propagate infected cuttings, then that's a common way in the nursery trade for the virus to spread. And you can also, like I mentioned earlier, have secondary spread within a crop if a population of thrips becomes numerous. But hopefully in most cases that won't be the case because they'll be detected and controlled. That's the hope. Okay, so I want to talk a little bit about some of the common hosts here. So in the case of ornamentals, I've listed a lot here. The two families, the Asteraceae family are probably contain the most susceptible hosts. And in the case of vegetables, it's the solanaceous vegetables that are common hosts. Okay, and also in some field crops like tobacco and peanut. These are what Tospoviruses are most commonly detected on. Symptoms, include stunting, mosaic patterns, which is when you have an alternation of infected and non-infected tissues, distortion, chlorotic streaking, yellow veins, okay, bronzing or reddening of the leaves, curling, and ring spots. And what I've done, I'm just going to go through quickly because I don't want to take up too much time. Should go through some symptoms. So this is chlorotic spots on young gloxinia. This is on geranium, and you can see here there's a close-up of some of these ring spots, which are often characteristic of tomato spotted wilt virus. This is some sort of mosaic pattern on begonia. You can see the alternation between infected and non-infected leaf tissues. This is Insp on chrysanthemum. You can see often that the flowers can also become deformed, and this sort of has like a burnt or frizzled type appearance. This is the same plant, but on the leaves here, you can see the symptoms on the leaves. This is on cyclamen. This is sort of a chlorotic spotting. This is on tomatoes, or tomatoes as they should be called. This is like a halo spotting, which is quite common on these fruit as a result of the virus. Pepper, here you can often see the fruit will not color up evenly. You get an uneven coloration on the fruit, and you can also see the symptoms on the leaves there as well. So this would obviously be unmarketable. This is field pepper. This is wilting. When these plants are infected at an early age, it almost looks like a vascular wilt. These kind of symptoms there, the entire crop can basically be completely eliminated. This is peanut. I know these aren't nursery crops, but these viruses were a huge issue in peanut. I think there were losses of over $100 million in Georgia in the 1980s, spread through western flower thrips and some of the other thrips. Okay, so in this state, this is some data from Kevin Ong here at the Texas Plant Disease and Diagnostic Lab. These are just counties in Texas that have submitted positive samples for Tospoviruses. In this case, these are all tested as positive for tomato spotted wilt or impatiens necrotic wilt virus. And you can see the list of the crops that were impacted there. I won't go through them all, but you can see there's quite a wide range of nursery and ornamental and vegetable crops. This is probably an underrepresentation because a lot of these samples get processed through private labs. They get processed on site, which I'll come back to quickly talk about how to do that. And they may otherwise go misdiagnosed. So we don't actually have a good handle on how common these viruses are, I don't think, in many cases. This is a slide that was provided by Debbie Groth-Helms, and she's the Director of Quality Manager at AgDia, which is one of the largest pathogen diagnostic labs, plant pathogen diagnostic labs in North America, probably the largest. And she provided this data to me, and these are some of the viruses that are currently most common in terms of the proportions of detections. Now they process thousands of samples per year. You can see Insp in the past couple of years is one of the becoming quite abundant. It's still a relatively low percentage of samples, but you have to appreciate that a lot of samples are being sent in as routine screening. So this is still represents an increase. The other ones we've got here, cucumber mosaic virus is also increasing. This is an aphid-borne virus. We've also got the Nemesia ring necrosis virus, which is another ornamental pathogen. The tomato ring spot virus, and yeah, that's it. These are actually either can be vectored by nematodes, so these are not thrips vectored, but I just wanted to share that data with you. Okay, just a few issues that I think are important to be aware of. One is that we're actually seeing new Tospoviruses now. Right now there's about 20 different Tospoviruses that have been described. About half of them have been accepted by the International Committee on Virus Taxonomy as officially Tospoviruses, but the others probably will follow suit. So the diversity is increasing. That's probably because a lot more people are looking for them now and describing them. We're also seeing an increase in the number of hosts that can be impacted. And interestingly enough, we're also seeing some new host-vector relationships. These Tospoviruses actually, they're very interesting. They can actually reassort their genome. Sometimes that happens when you get multiple infections in insects, and these Tospoviruses can actually mutate and reassort and lead to new species or new strains. And this seems to be an issue. And another issue we're having is that we're having various invasive thrips coming into the country. And so with this further enables this group of diseases to become more diverse. And we're also seeing some issues of resistance-breaking strains. This is mainly an issue with vegetables. There's been a lot of breeding for resistance with vegetables, but we're now seeing insect and Tospovirus systems that are actually overcoming resistance. So this is kind of an ongoing battle to try to stay on top of these issues. So just really quickly, what ones do we have in North America? We have, as I mentioned earlier, tomato spotted and impatiens necrotic wilt viruses they've really been here since the 1980s and the 1990s that we know about. More recently, we're seeing Irish yellow spot virus. Now that was detected in the 1990s, and that one is really being mainly an issue on onion. It's a big issue in the Northwest. We also have one on soybeans, the soybean vein necrosis virus. That was discovered in 2008 but described in 2011. That's again a major issue, but that seems restricted to soybeans. And then these final two, the groundnut ring spot virus and tomato chloric spot virus, these were more recently discovered. They're really issues, so far they seem to be primarily issues in vegetables, but we're still not fully sure of their host range. So the idea that these could become an issue in the nursery trade is something that we need to kind of keep our eye on. There's also another group of viruses, the alar viruses, that are also starting to become more prominent. There's a couple here. There's the tobacco streak virus, which is the type specimen. So that's the one that was first described. And in 2013, there was a group in Florida detected a related one, the tomato necrotic streak virus. And I know that these viruses are being looked at a lot in the lower valley where there's a lot of vegetable production. So as of now, that's the status. But there's but one of the issues we have, and I apologize, this slide's not very clear, and it's also a little out of date. But one of the issues we have is if you look worldwide at the other strains of these viruses we have, probably the second greatest diversity are found in South America. And so, you know, in Texas here, we're very much in the firing line for a lot of these viruses to come in. We have at least 5 or 6 other ones that have been described, and probably many more that haven't been described, which can impact vegetable and ornamental plants. So we're going to be seeing more of these viruses come in. One of the issues we have is that we don't necessarily always have the tools to detect them. So this is one of the things I'm interested in doing. Just moving on a little bit here, I was talking about some of the new invasive thrips which might spread the Tospoviruses. This is one that you should be aware of if you're not. This is the chili thrips. And I'm actually giving a webinar on this in next month. This is an insect that's very close to my heart. It's established in Texas, and a few years ago, and it seems to be starting to spread. So this is another issue that I'll talk about in a few weeks. I was at a meeting a few weeks ago in Dallas, the Northeast Texas Nursery Growers Association, and found out about another invasive thrips there. This is the Japanese flower thrips. This was actually detected on hostas in Michigan, and I believe that it's currently been eradicated. But this is just another example of how these thrips do come in. And this is actually a virus, Tospovirus competent species. So again, as new species come in, they can spread viruses to potentially new hosts. Okay, so what to do about it? This is probably the main part of the talk. I apologize for taking so long to get here, but because the symptoms are so variable, it's very hard to get a diagnosis. So typically, growers use serological assays. These are assays that rely on antibodies that can capture the specific virus proteins and then conjugate a reaction to indicate that the virus is present. So ELISA would be, if you're not familiar with ELISA, ELISA is a relatively simple diagnostic test that you can do, but you do need to do it in a lab. And these kits have been available for a while. But again, because they need a certain basic laboratory equipment, they take a few days to do. They're not necessarily something that can be done on site. More recently, we have an improvement in the technology through the use of immunostrips. These are strips that allow testing in the field, and I have a slide on that. And other ways to do diagnosis would be molecular tests. So typically PCR, you can either use broad or relatively conserved primers, and we can use that to detect a whole range of Tospoviruses, but then might need to be sequenced in order to determine what they are, and that's how the new species are being discovered. There are a couple of testing services. So Agdia, I mentioned earlier, and Bioreba, they both have a testing service, and they also sell equipment for home testing. This was a slide that was shared, it was given to me by Debbie at Agdia, so I wanted to show that. There's some information there where you can purchase the equipment. You have, this is an immunostrip or a dipstick, I think some people call them, on the left here. So these contain the antibodies and the reagents on the strip. You can see on the lower part of the strip there. So in this case, you can just take a piece of suspected plant tissue, grind it up in a little bag, and they will provide a bag with the buffer in, and you just put the end of the strip in there, and if the virus protein is detected, it will conjugate a reaction which shows us a little strip. It's kind of similar to a pregnancy test kit. So these are relatively easy, and you can get results within a few minutes as opposed to a few days if you're shipping samples or doing the testing in the lab. So this is a very useful technology, and these strips have to be specific for specific pathogens, not just Tospoviruses. They're also available for bacterial pathogens and other things as well. So, okay, I want to talk very briefly about thrips management because there's no cure for the Tospoviruses, so management's obviously important, and this is probably another talk in itself, but here's just some general principles that really are critical when it comes to maintaining clean operations. So starting clean is always very important. You know, if you don't start clean, then you're going to have a problem to begin with. So inspection of new plants coming in, if necessary, quarantining, conducting routine testing to determine, to find out if there are any issues and whether any management needs to occur. This is important. Using virus-free plants. I mentioned how the life cycle of the Tospovirus works. So if you can remove weedy hosts both inside a greenhouse or outside and in a nursery situation, that goes a long way to removing sources of the virus that can come in. Screening is very important, and I'll talk a little bit about that. Monitoring is important, and then you've got a whole range of chemical and biological controls, which I'll just touch on briefly. Okay, so insect screening, I'm sure that those of you that have greenhouses or work in greenhouses are familiar with the need to screen, especially from the intake side of both the intake side and the outtake side for fans. This is obviously probably one of the commonest sources of insects getting into greenhouses. Vents, windows, doors, these are all areas where you can have access ways to thrips that can be carrying, or other insects indeed that can be carrying diseases that you don't want in your operation. Here you have just a slide of insect screening on fan equipment here. Now there are different sizes of insect screening, and thrips are amongst one of the smallest, but I should point out that thrips can get through insect screening. I've seen that happen, so this is not 100% guarantee. Monitoring is important. Thrips are attracted to blue. They're one of the few insects that like blue, so sticky cards are a good way to monitor. These need to be placed strategically, so near doors, near vents, near areas where people are working, along aisleways, anywhere where people are moving and things can be brought in. So these are insecticides that are registered for thrips control on ornamental plants in Texas. Currently, some of them only have a greenhouse label. And you can see some of these materials work quite well. I've tested some of them in the past. If you're interested, feel free to email me. There's some data on some of these. But insecticides are notoriously unreliable against thrips for various reasons, and thrips are also able to develop resistance to materials. So relying on an insecticide-only program is probably not a good idea. You should really do everything to minimize the pressure in the greenhouse as well. There's a number of good biological controls for thrips. I know I'm getting off topic a little bit here, but in Florida, a lot of growers are now relying exclusively on biological control methods to control thrips, and they're having a lot of success. But you do need to have a rigorous program for that. So if anyone has any questions, or feel free to follow up with me, and I can hopefully point you in the right direction with that. These are just some growers that are using biological control for thrips that are vectors of Tospoviruses. The guy on the top here, he's just treating a local hotspot. This is in Majesty Ponds. And on the bottom screen, you can see that they're applying the biological controls over a larger area using an air blast system. Okay, so Mung asked me to keep it to about half an hour, so I wanted to do that. I probably skipped some things a bit quickly, so I didn't know if there's time for questions. I'm happy to answer any questions. But here's some resources here, some resources for testing sites. And there's some good articles that I've highlighted there that really document in more detail how integrated pest management programs for thrips work, with a focus on protecting against Tospoviruses. So I think I will end it there. I want to thank everyone for their attention. I hope you could hear me. And if there's any questions, I'd be happy to answer them. Thank you, Meng Meng.